Hello! In this session, we're going to talk about ancestor DNA reconstruction, why it's important, how it can be done, and tools that exist for doing it. As with all RootsTech Connect talks, the views in this session are those of myself and do not necessarily represent Family Search International or RootsTech. Humans have 23 pairs of chromosomes, where they inherit one copy of each chromosome from their father and one from their mother. A picture of a human male's 23 pairs of chromosomes is shown here. This is a male because this person has a Y chromosome. Highlighted here from chromosome 3 is one copy outlined in blue and one in red to represent that this person has a copy from their father and one from their mother. Throughout this talk, we'll see figures like this, where squares represent males, circles represent females. This horizontal line means this is a couple, and these are their 4 children. Below each shape are 2 vertical bars of different colors that represent the 2 chromosomes of, for example, chromosome 3, or any of the other 22 autosomes. The sex chromosomes are handled differently, and we won't talk about those. Now, this father has a dark blue and a light blue chromosome, and as you can see, each child inherited a chromosome from their father and also one from their mother. But these children's chromosomes are mosaic. They're joined together pieces of these 2 chromosomes, where this second child inherited perhaps 75% of this chromosome from the light blue copy from his father, and the remaining portion from the dark blue copy. And all the other children inherited different proportions, as you can see. Now this process of recombination is important to keep in mind when we think about trying to reconstruct a person's DNA. Now all of our DNA comes from our 2 parents, and their DNA comes from their parents, and so on back through time. If we were able to identify which ancestor a segment of DNA came from, we could use that DNA to partially reconstruct that ancestor's DNA. So as an example, these 3 individuals, I should mention that in this talk, individuals whose shapes are not filled are those that we don't have DNA directly for. These 3 individuals at the bottom, we do have DNA directly for, and each of them has inherited a dark blue piece of DNA from their grandfather. Now, unfortunately, the DNA isn't color-coded, so we can't immediately apply this sort of principle in practice, but if we were to be able to somehow identify that these segments of DNA came from the grandfather, then we could reconstruct that grandfather's genome. So as a definition, throughout this talk, we will say that an ancestor's DNA is reconstructed when we have determined as much of the DNA as we can from that ancestor's descendants. So now let's talk about why it's important to attempt to reconstruct DNA from our ancestors. So a bit of background. Many of you know about shared segments. These are also called identity by descent segments. If you haven't heard of this before, these are regions of DNA, such as at the beginning of this chromosome in this first and second individual at the bottom, that they've inherited from a common ancestor. Here in red, this individual has inherited from their grandmother their DNA, and so has this individual. Therefore, they have identical DNA across that stretch at the beginning of this chromosome, and this can be detected. And this is what genetic testing companies use to report relationships. Another point to keep in mind is that a child inherits a random half of their parents' DNA, and this is useful for reasons we'll talk about. Perhaps the most important reason why it's useful to reconstruct DNA from our ancestors is that our ancestors, on average, share more DNA with our relatives in these shared segments or IBD segments than we do. This is because of the fact that a child, in this case us, only inherits half the DNA of our parent. And therefore, on average, the child, again us, will only inherit roughly half of the shared segments that our parent has with this other individual. Now, I should emphasize that this is only on average. There are times when a parent might transmit all of their segments to us. In that case, reconstructing DNA from our parents is still useful because it helps us realize that the rest of our parents' DNA actually doesn't contain any shared segments. So an implication of what we just talked about is that it's actually easier to determine how closely related two individuals are when they are more closely related than when they're not. So, for example, my parent is more closely related to my second cousin than I am. So it's easier to determine their relationship. We'll see that in these plots. But before we go through them, I'll mention that these tools that are being used for these slides are those that are commonly used in academia, but companies use similar tools. Companies tend to report ranges rather than precise relationships, partly for the reasons that are shown here. But the key point is that it's easier to more precisely determine the relationship type of individuals that are more closely related, that is, from my parent or perhaps my grandparent to a distant relative than from me. So on this first slide, we see that from real data, this is academic data, not generated by genetic testing companies, but similarly processed, full siblings can basically be reliably determined 100% of the time. By contrast, first cousins, if we're going to try and precisely classify how closely related these individuals are, almost 6% of first cousins will look either more closely related, such as looking like they're half siblings, or less closely related, such as looking like they're first cousins once removed. That's quite a drop, but an even more dramatic drop is from first cousins to second cousins, where the relationships for second cousins are only reliably determined in about 68% of cases. So other cases tend to be inferred as first cousins once removed or second cousins once removed. Now again, it's important to realize that companies don't tend to report precise relationship types, but the potential is there for us as individuals to try to analyze the data and to be able to be much more confident in the results. So if two second cousins got tested, but then also had their parents tested or their parents reconstructed, they would move from having a 68% reliable classification rate to a 94% classification rate. Another reason why it's good to reconstruct our ancestors' DNA is that part of the goal of genetic genealogy is to figure out how we're related to each other. Ultimately, we'd like to figure out which common ancestors all of our relatives are related to us through. But if we can't go that far, at least we could start to go part way there. We could say that, for example, this person on the far left is related through our maternal grandfather, and the person on the right through our paternal grandmother, and so on. So reconstructing DNA of our ancestors allows us to do this. So in summary, it's important to reconstruct DNA from our ancestors because our ancestors share more DNA with our relatives than we do, and therefore can enable more reliable, more precise classification of how our ancestor and that person are related. Secondly, it's good and moves towards our goal in genetic genealogy if we can figure out how we are related to people, and it may also help our relatives to figure out their genealogy if we can tell them who they're related to us through. We can give them places and names associated with those people. So now that we've motivated ancestor DNA reconstruction, let's talk about how it works. So we'll talk about two basic approaches for doing this. In the first, we have data for one parent and some number of children, and we want to reconstruct the DNA of a missing parent. So shown here is a case where we have DNA from a dad and 4 children, but we need DNA from a mother. Now, the beauty and the benefit of the fact that each child inherits a random half of the parent's DNA is that we can get different snapshots, different segments of DNA from each child. In fact, it's useful to have more children for this reconstruction. You can get more DNA from the parent when you have more children. And this works simply by, in some sense, subtracting away the DNA that, in this case, the father transmitted to these 4 children, and then joining together the segments of DNA that the mother therefore must have transmitted into her pieces of DNA, her chromosomes. Now, the second method is a bit more complicated. Unfortunately, if we have, for example, a set of siblings, but neither parent, or, for example, some distant cousins, maybe even first cousins, who are related through 2 common ancestors, unfortunately, we can't know whether these shared segments come from the common father or grandfather or the common mother or grandmother. So the approach that is taken to deal with this is to take DNA from other relatives that we know are not descended from these ancestors, but are related to them. So here is shown a picture of a set of siblings that have, of course, a father and a mother, and we have relatives through the father and through the mother. And we can use the shared segments that these siblings have in common with these other relatives to piece together the DNA from their common parents. Now, this is a great approach, and it works really well. And at the same time, it can be laborious to determine who is related to these ancestors but not descended from them. And we can only be confident of DNA that has a shared segment with these other individuals. So we don't always get a lot of DNA from this approach, but it can be very, very useful. And even though this picture shows a set of siblings and other relatives, this same principle applies for the cousin case. All we need is individuals that are related to the common ancestors but not descended from them. So lastly, tools that do these forms of reconstruction. The first method is done by MapS and Happy. Happy is the subject of another talk here at RootsTech Connect. The title is on this slide. And then this second approach is done by Lazarus at GEDmatch or by Borland Genetics. And so I encourage you to check these tools out and try to make use of them. In this other talk, there's information about Happy, about the accuracy. This first method can do a very reliable job and a very complete job of reconstructing the parent's DNA. So feel free to check that out. And with that, I want to thank Debbie Kennett, who provided a lot of information that was useful as I was looking at tools that are available for doing reconstruction. Also, John Blangero and the team that has collected the data for the San Antonio Mexican-American Family Studies. That was the real data that I was mentioning earlier, and there's a little more details on that data given in this other talk. I also want to thank those that have helped fund this work, and thank you for listening.